For this topic, the salivary glands, we're going to tackle the general structure, secretory cells, myoepithelial cells, duct system, the classification, the development, the functions of saliva, and the clinical considerations. Salivary glands are organs which synthesize and secrete their secretions over an epithelial surface via a hollow channel. These glands are present in and around the oral cavity, and its secretions play an important role in the physiological processes of the oral cavity. One of the key players here is first the secretory cells arranged in acini, which may be tubular or grape-like. Secretory cells that are making up one acinus opens up into a lumen, which is its central cavity. The lumen of several acini are connected with one another by a secretory tubule, which in turn will lead to the duct system of the gland. An acinus consists of several secretory cells. Then, a collection of several acini is called a lobule. A group of lobules is called a lobe. This illustrates the hierarchy in salivary glands. Salivary glands consists of a group of lobes, which consists of lobules, then acini, then down to secretory cells. The duct system also plays a key role in producing saliva. The secretory units merge into intercalated ducts, which is collectively called the duct system, which are lined by CT, simple low cuboidal epithelium, and surrounded by myoepithelial cells. This system actively participates in the production and modulation of saliva. The flow of saliva excretion is from lumen of secretory tubule to intralobular duct to interlobular duct to main excretory duct, then lastly to oral cavity. This shows the basic structure of the duct system. As what was previously stated in the duct system, a connective tissue surrounds both the secretory cells and the duct system. It demarcates the gland from adjacent structures. The connective tissue capsule has extensions that go into the body of the gland, which divides salivary glands into lobes and lobes into lobules. Again, connective tissue capsule surrounds the salivary glands. Septa from the capsule divide the glands into lobes and lobules and also carry a larger arteries and veins. The salivary glands are compound tubulo-acinar glands, which means they have branched ducts ending in sac-like dilations, acini, where the excretory cells are located. The secretory unit of the salivary gland consists of an acinus, intercalated duct, striated duct, and excretory duct. The connective tissue capsule consists of first blood vessels. Blood vessels follow the course of the duct up to the level of lobules, and when it reaches the acini and secretory cells, it forms a capillary network surrounding the acini. Second is the nerves. Each salivary gland has a sensory or afferent nerve, two motor or efferent nerves. Both parasympathetic and sympathetic nervous system innervate this structure. So, the enzymes and the mucus are produced by two distinct cell types called serous cells and mucus cells. Serous cells synthesize, store, secrete proteins and glycoproteins. Their secretion is watery and is rich in proteins and enzymes. Secretions in the secretory granules are discharged into the lumen by exocytosis. The fusion of the secretory granule membrane and the cell membrane is followed by the rupture and release of the product. Integrity of cells maintained as only as the secretion is released. The secretion consists of zymogen granules, precursor of amylase, primary enzyme found in saliva functions for breakdown, digestion of CHO. Breakdown of CHO is not so extensive because contact time between saliva and food is very short. CHO, carbon, hydrogen, oxygen, or aldehyde. The second type of secretory cell is the mucus cell. Mucus is usually arranged in a tubular and or polygonal configuration with a larger central lumen. They are pyramid-shaped with a flattened nucleus at its base because of the presence of the secretory droplets which push the nucleus. They are classified under cuboidal cells, and at the base, you can find the flattened nucleus and other organelles. The nucleus is flattened because of the presence of the secretory droplets which push the nucleus. Its apical cytoplasm does not stain intensely and contains secretory product. If serous cells synthesize, stores, and secrete proteins, mucous cells synthesize, stores, and secrete mucin. Mucin contains more CHO than protein. Mucin is secreted to lubricate and protect the oral mucosa. It forms a barrier on surfaces and bind and aggregate microorganisms. They are thick and more viscous than the serous secretion. Mucin is discharged by fusion of membrane of secretory droplets to apical plasma membrane. As it is discharged, a part of mucous cell goes with the mucus secretion. Here, you can see that the serous cells are pyramidal or triangular in shape, while the mucous cells are columnar in shape. For serous acini, it's triangular in shape and appear darkly stained with the HE. The basal portion of the glands appear darker stained due to the presence of rough endoplasmic reticulum and the round basally located nuclei in the cells, while the apical portion stains lighter due to the presence of secretory granules. Mucous acini can easily be distinguished from serous acini, as the cells of the mucous acini are paler and contain flattened nuclei located toward the base of these cells. Here, you can see how the secretory cells and duct system are connected. Myoepithelial cells, also known as basket cells, are contractile cells associated with the secretory end pieces and intercalated ducts of the salivary glands. These cells are located between the basal lamina and the secretory acini or acinar cells. They are joined to the adjacent ductal or acinar cells through desmosomal junctions or desmosomes. These cells have a flat nucleus and a small cell body, from which 4 to 8 processes branch out. These processes, or extensions, surround the entire secretory unit, called the acinus. In fact, they are also called the basket cells because they resemble a basket holding secretory cells. Myoepithelial cells associated with the ductal cells, on the other hand, have a more fusiform shape with fewer processes compared to those that are present around the secretory end pieces. Their cytoplasmic processes contain the myofilaments, actin and myosin, similar to smooth muscle cells. The image shows a transmission election micrograph of a myoepithelial cell at the base of a mucous secretory cell. The arrows point to the cytoplasmic processes containing the myofilaments. Myoepithelial cells have the following functions. They act as a support for the secretory cells, preventing overdistension. The main function of the myoepithelial cells is to contract and squeeze out secretory products that accumulate within the cytoplasm of the acinar cells. They help expel the primary saliva from the end piece into the duct system. Second, they contract and widen the diameter of the intercalated ducts, lowering and raising the resistance to outflow. They also provide signals to the acinar secretory cells that are necessary for maintaining cell polarity and structural organization of the secretory end piece. According to research, myoepithelial cells also have a protective function. They secrete proteins with tumor suppressor activity, which prevents the growth of cancer cells, and anti-angiogenic activity that inhibits the blood vessels, which might be a source of nutrition for cancer cells. The ductal system of salivary glands is a varied network of tubules that progressively increase in diameter, beginning at the secretory end pieces and extending to the oral cavity. It is more than just a simple conduit for the passage of saliva. It actively participates in the production and modification of saliva. The structure of the salivary glands can be likened to a bunch of grapes. Grapes are the secretory end pieces, and the stem or branches are the ducts. There are 3 classes of ducts, each with differing structure and function. First is the intercalated duct. It is the smallest intralobular duct. It drains the acinus of the salivary gland. This is where the saliva from the secretory end pieces first passes through. The intercalated ducts are lined with simple cuboidal epithelium and myoepithelial cell bodies with processes located along the basal surface of the duct. They can modify salivary secretions by the addition of macromolecular components stored in their secretory granules. These macromolecular components are known to have antibacterial properties, which occurs in various ways. Lactoferrin, for instance, binds free iron, thus depriving the bacteria of its essential element. Meanwhile, lysosomes have the ability to hydrolyze bacterial cell walls. In the image, the arrowheads point to the branching intercalated duct joined by several serous end pieces in the human submandibular gland. The duct cells are low cuboidal, and their cytoplasm stains lightly with eosin. Located at the right is an image of an intercalated duct cell. Next is the striated duct. Striated ducts constitute the largest portion of the duct system. It is also an intralobular duct similar to the intercalated duct. Their cells are columnar, with a centrally placed nucleus and pale acidophilic cytoplasm. The next image shows a light micrograph of striated ducts and the human submandibular gland. The ducts have large lumina and are lined by a pale staining simple columnar epithelial cells with centrally placed nuclei and faint basal striations. Radially oriented lines or striations may be observed in their basal cytoplasm, hence the term striated. These lines are actually enfoldings of the basal membrane. In between these lines, a large amount of mitochondria are located. These mitochondria provide the energy needed for active transport of substances. On the right is an image of the basal region, with visible enfolded membranes located in between the mitochondria. Striated ducts modify the saliva by the reabsorption of sodium and chloride ions and secretion of potassium and bicarbonate ions. During the process, more substances are being absorbed than secreted, thus making the saliva hypotonic, having low concentration of Na, Cl ions, and increased concentration of carbonate and potassium ions. Lastly, we have the excretory duct. In comparison to the previous two, it is classified as an interlobular duct. Excretory ducts are located in the connective tissue septa between the lobules of the gland. They have pseudostratified epithelium with columnar cells extending from the basal lamina to the ductal lumen. The epithelium of the main excretory duct, however, becomes stratified upon reaching the oral opening. Small basal cells are also present on the basal lamina. Unlike columnar cells, they do not reach the lumen. Similar to striated ducts, they can also alter the electrolyte concentration of saliva. Mitochondria and enfolded basal membranes are also present in the cells of excretory ducts. But as they increase in size, the number of mitochondria and the extent of enfoldings of the basolateral membranes decrease. The following cells are also present in the excretory ducts. Mucus or goblet cells that are mainly found in large excretory ducts. Tuft or brush cells, which are believed to be a type of receptor cells. Lymphocytes, macrophages, and dendritic cells, which play an important role in human surveillance. The image on the left shows a similar excretory duct in the interlobular connective tissue. The duct epithelium is pseudostratified with tall columnar cells and a few basal cells. The arrowheads point to the numerous capillaries and venules present around the duct. Meanwhile, the second image shows a large excretory duct surrounded by dense connective tissue. The pseudostratified epithelium contains several mucous goblet cells specified by the arrowheads. Human salivary glands are classified into two groups. First is according to nature of the secretion. Under this, we have serous glands that consist of purely serous secreting cells. Mucous glands consist of purely mucus secreting cells, while mixed glands, based on its name, consist of serous and mucous cells. In the figure, the serous part is a crescent shape called serous demilons or demilons of Giannuzzi. Second, classification of human salivary glands is according to size and location. And then under it, we have major glands and minor glands. Major glands are large and can be found outside the oral cavity. It can also be found on both left and right side of the face. It also has a main excretory duct that is so long it is able to reach the oral cavity. And it contains the general structures of the salivary gland, namely dense connective tissue capsule, duct system, and secretory cells. The first major gland we have here is the parotid gland, the largest among all of them. It is located subcutaneously on either side of the face, anterior and below the ear, and it extends from zygomatic process to the angle of the mandible. Its main excretory duct is Tenson's duct. Its opening is marked by the parotid papilla. Then it is a pure serous gland. Blood here is supplied by internal carotid artery, and it is innervated by cranial nerve 9. The parotid gland secretes watery saliva that is rich in enzymes such as amylase, proteins such as proline-rich proteins, and glycoproteins. A submandibular gland is classified as a mixed gland because it contains both serous and mucous cells. Although the proportions of serous and mucous secretory end pieces may vary from lobule to lobule, and among individual glands, serous cells significantly outnumber the mucous cells. It is located in the submandibular triangle, behind and below the free border of the mylohyoid, inner surface off angle of mandible, situated in the posterior part of the floor of the mouth, adjacent to medial aspect of mandible. Its main excretory duct is the Wharton's duct, which opens on the floor of the mouth, at the summit of the sublingual papilla, also called the carnuncula sublingualis, lateral to the lingual frenulum. This gland is innervated by the cranial nerve 7 and is supplied by the facial and lingual arteries. It also releases saliva, which contains highly glycosylated substances called mucins. These substances contribute to the different functions of saliva. This is a light micrograph of a submandibular gland. Serous secretory end pieces pointed by the yellow arrows predominate, but a few mucous tubules pointed by the green arrows are also present. Among the 3 major salivary glands, the sublingual gland is the smallest. It is also a mixed gland, but mucous secretory cells predominate. It has one main duct with various small ducts. The main duct, Bartholin's duct, opens with or near the Wharton's duct, while the smaller ducts, called the ducts of Rivinus, open independently along the sublingual fold. Similar to the submandibular gland, it is innervated by cranial nerve 7 and is supplied by the sublingual and submental arteries, which arise from the lingual and facial arteries, respectively. It also produces viscous saliva that is rich in mucins. The image shows a light micrograph of a sublingual gland. Mucous tubules, identified by the yellow arrows, are abundant compared to the serous cells pointed by the green arrows. Aside from the major salivary glands, there are also about 600 to 1,000 minor salivary glands located just beneath the epithelium in the oral cavity. They lack a distinct connective tissue capsule, and they only have short ducts that open directly into the mouth. These glands are classified based on their location in the oral cavity. First, labial and buccal glands. These are the minor salivary glands found in the lips and cheeks. They are a mixed gland, but are predominantly mucous. Their openings are scattered all over the surface of the lips and cheeks. Second, palatine glands. They are purely mucous glands located in the posterolateral zone of the hard palate. They are also found in the submucosa of the soft palate and the uvula. Third, lingual glands. They are divided into two, the anterior lingual and the posterior lingual glands. The lingual glands found at the anterior are also called the glands of Blandine and Nunn. Serous and mucous cells are both present, but mucous cells predominate. They are found near the tip of the tongue, and their ducts open into its ventral surface. Fourth, posterior lingual glands, and there are two types: the glands of root of tongue and the glands of von Ebner. The glands of root of tongue is purely mucus. The glands of von Ebner, on the other hand, are purely serous. Ducts of the glands of root of tongue open into the dorsal surface of tongue, while the glands of von Ebner open into the canal surrounding the circumvallate papilla. In the development of the salivary gland, the parotid glands begin to develop at 4 to 6 weeks of embryonic life, the submandibular glands at 6 weeks, the sublingual and minor salivary glands at 8 to 12 weeks. Similar to teeth, the individual salivary glands arise as a proliferation of oral epithelial cells, forming a focal thickening that grows into the underlying entomesenchyme. Continued growth results in the formation of a small bud connected to the surface by a trailing cord of epithelial cells, with mesenchymal cells condensing around the bud. Mesenchyme will give rise to the connective tissue capsule of the gland. Proliferation of the epithelium into the underlying mesenchyme results in long epithelial cords, which are pointed by arrowheads. These cords undergo repeated dichotomous branching. The mesenchyme here, MES, has condensed around the developing glandular epithelium. Clefts develop in the bud, forming 2 or more new buds. Continuation of this process, called morphogenesis, produces successive generations of buds and a hierarchic ramification of the gland. The string of cells will branch. The development of a lumen within the branch epithelium generally occurs in this order: 1. In the distal end of the main cord and end branch cords; 2. In the proximal end of the main cord; and 3. In the central portion of the main cord. Lumen formation, shown by arrows, has begun in the ducts. Branching of the distal ends of the epithelial cords is evident, as shown by arrowheads. After development of the lumen and the terminal buds, the epithelium consists of 2 layers of cells. The cells of the inner layer eventually differentiate into the secretory cells of the mature gland, mucus or serous, depending on the specific gland. Some cells of the outer layer from the contractile myoepithelial cells that are present around the secretories and pieces and intercalated ducts. As the epithelial parenchymal components increase in size and number, the associated mesenchyme connective tissue is diminished, although a thin layer of connective tissue remains, surrounding each secretory and piece and duct of the adult gland. Thicker partitions of connective tissue, septa, continuous with the capsule and within which run the nerves and blood vessels supplying the gland, invest the excretory ducts and divide the gland into lobes and lobules. The following are the functions of saliva. First on the list is protection. Saliva protects the oral cavity in many ways. The fluid nature of saliva provides a washing action that flushes away non-adherent bacteria and other debris. In particular, the clearance of sugars from the mouth limits their availability to acidogenic plaque microorganisms. The mucins and other glycoproteins provide lubrication, preventing the oral tissues from adhering to one another and allowing them to slide easily over one another. The mucins also form a barrier against noxious stimuli, microbial toxins, and minor trauma. Second on the list is buffering. The bicarbonate and, to some extent, phosphate ions in saliva provide a buffering action that helps protect the teeth from demineralization caused by bacterial acids produced during sugar metabolism. Some basic salivary proteins also may contribute to the buffering action of saliva. Additionally, the metabolism of salivary proteins and peptides by bacteria produces urea and ammonia, which help to increase the pH. Third is carbohydrate digestion. Saliva also contributes to the digestion of food. The solubilization of food in substances and the actions of enzymes such as amylase and lipase begin the digestion process. The moistening and lubrication properties of saliva also allows the formation and swallowing of food bolus. Next is taste sensation. Saliva functions in taste by solubilizing food substances so that they can be sensed by taste receptors located in taste buds. Saliva produced by minor glands in the vicinity of the circumvallate papillae contains proteins that are believed to bind taste substances and present them to the taste receptors. Additionally, proteins in saliva, specifically gustin, have a trophic effect on taste receptors and are involved in growth maturation of taste buds. Next is the antimicrobial function. Saliva has a major ecological influence on the microorganisms that colonize oral tissues. These are factors which enable it to create an environment in the oral cavity which is unsuitable for microbial growth. In addition to the barrier effect provided by mucins, saliva contains a spectrum of proteins with antimicrobial activity such as the lysosome, lactoferrin, peroxidase, and secretory leukocyte protease inhibitor. The major salivary immunoglobulin, secretory immunoglobulin A, IgA, causes agglutination of specific microorganisms, preventing their adherence to oral tissues and forming clumps that are swallowed. Next on the list is maintenance of tooth integrity. Saliva is supersaturated with calcium and phosphate ions. At the tooth surface, the high concentration of calcium and phosphate results in post-eruptive maturation of the enamel, increasing surface hardness and resistance to demineralization. Last on the list is tissue repair. A variety of growth factors and other biologically active peptides and proteins are present in small quantities in saliva. Under experimental conditions, many of these substances promote tissue growth and differentiation, wound healing, and other beneficial effects. The following are known clinical considerations associated to salivary glands. Xerostomia or dry mouth is a common clinical complaint. Oral dryness occurs most commonly as a side effect of medications taken by the patient for other problems. Loss of gland function occurs after radiation therapy for head and neck cancer because the salivary glands often are included in the radiation field, and salivary gland cells are highly sensitive to the deleterious effects of radiation. Chemotherapy for cancer or associated with bone marrow transplantation may also cause reduced salivary function. Autoimmune diseases, in particular Sjogren's syndrome, may cause progressive loss of salivary function from the invasion of lymphocytes into the gland and the destruction of epithelial cells. The decreased volume of saliva in the mouth leads to drying of the oral tissues and loss of the protective effects of salivary buffers, proteins, and mucins. The oral tissues are more susceptible to infections and speech. Eating and swallowing become difficult and painful. The teeth are highly susceptible to caries, especially near the gingival margin. Temporary relief is achieved by frequent sipping of water or artificial saliva. Patients who have some functional salivary tissue may benefit from pharmacologic therapy with oral parasympathomimetic drugs such as pilocarpine to increase salivary flow. Sialolithiasis and mucocele or mucus retention cyst. Blockage of a duct may cause a transient swelling associated with eating as blood flow increases and saliva backs up in the gland. Ductal obstruction may result from the formation of sialoliths, stones. Most common in the submandibular duct or a mucus plug or the severing of the duct of a minor salivary gland by trauma. Pathologic changes in salivary glands are also observed in individuals with cystic fibrosis. Salivary sodium and chloride concentrations are increased, and mucus-secreting glands may develop mucus plugs. Mumps, viral infections such as mumps and bacterial infections of individual glands may cause inflammation resulting in a painful swelling. Age changes. With age, a generalized loss of salivary gland parenchymal tissue occurs. A gradual reduction of up to 30% to 60% in the proportional acinar volume of the major salivary glands has been observed. The lost salivary cells often are replaced by adipose tissue. An increase in fibrous connective tissue and vascular elements also occur. Changes of the duct system, including an increase in non-striated intralobular ducts, dilation of extralobular ducts, and degenerative and metaplastic changes, have been reported. Thank you for listening.